Hello, Mr. Jarrett. My name is Kareem El Shatori, and this is my video response to my what if questions. So, I received two what if questions. My first one was, what if the temperature around a C3 plant was raised to 40 degrees Celsius for an hour and then again to 60 degrees Celsius for 3 hours? Also, I received another one, and that was, what if NAD plus reductase was removed from all the thylakoid membranes in a plant? So I hope you enjoy this video, and thank you very much. So in this first what if question, first let's talk about what exactly a C3 plant means. So it's simply C3 plants are the majority of the plants and crops that we know of, and the name is referring to the carbon compound that is produced in its photosynthesis. So usually with these type of plants, they function best at temperatures around 0 to 30 degrees Celsius, and they kind of thrive in this zone. But the main question is, what happens if these temperatures increase? How does this change the plant? So to begin this discussion, a very important thing must be mentioned, and it's in this case an enzyme, and this enzyme is called RUBP. So specifically, RUBP is very important in the Calvin cycle. So think of RUBP as like a pet dog. So a pet dog is usually with its owner and around its owner. So just like that, RUBP usually incorporates CO2 in its first stage of the Calvin cycle. But also sometimes like a pet dog, the pet dog can stray away from its owner or run away from home, etc. So in this stage, once in a while, RUBP might stray away chasing anything else other than CO2, and this can be an oxygen atom. So instead of incorporating CO2 to complete a successful Calvin cycle and create one glucose molecule, sometimes RUBP snatches an oxygen atom, and at the end, the Calvin cycle does not make a glucose. So it also ends up wasting carbon atoms, which has not a very meaningful effect for photosynthesis. And instead, actually, it's called something like photorespiration. So, what we need to realize or ask ourselves is, so why does RUBP do this? Why can it choose a CO2 molecule instead of an O2 molecule? And in the first stage of this question, it says that the plant will increase to 40 degrees for 1 hour. So, when a plant has its stomata, or basically, I'm talking about the leaf pores, they open and they close. So, when they open, CO2 diffuses in, while water, vapor, and O2 molecules diffuse out. This is done to basically minimize photorespiration, as I said before, and keep the Calvin cycle flowing normally. However, when temperatures increase, the plant tries to minimize this behavior to avoid water loss and instead results in a buildup of oxygen atoms. So that throws off the ratio of carbon dioxide to oxygen inside the plant, and it creates this issue. So, also something to take note of is that RUBP has a higher affinity for oxygen when temperatures increase. In the first hour of the plant, in this case, the RUBP will bind to CO2 molecules normally about 80% of the time, since it's minimal temperature increase than the normal zone. But usually in this scenario, the bigger issue happens in the second stage where the temperature increases to about 60 degrees Celsius. So in this case, RUBP's affinity for oxygen increases a lot more and will tend to grab an oxygen atom more often than a carbon dioxide. So to get down to the real details, in the first stage of the Calvin cycle, as you can see, two molecules are created: a three carbon molecule, which is called 3-PGA, and another molecule called phosphoglycolate. So 3-PG is normal in the Calvin cycle when RUBP grabs a carbon dioxide, and uh, but this other atom called phosphoglycolate is not. So therefore, that molecule cannot enter the cycle, so it's left out. So therefore, all the carbons in the molecules are essentially they're essentially wasted and redirected through other organelles to break it down. So, more importantly, though, this means that there will not be enough G3P in the cycle to make one glucose molecule, which is the main objective of photosynthesis. So this means, what does this mean? So this means for the 3 hours where the temperature increases to 60 degrees, the plant will not be able to make its own food. Essentially, these 3 hours, the plant will go through the process of starvation. Some research shows that this is kind of like a defense system and is done by plants in order to protect light damage from molecules involved in photosynthesis. In my second what if question, I was asked to consider what would happen if NADP plus reductase was removed from all of the thylakoid membranes. So first, let's look at what is a thylakoid. So as we can see from this image, a thylakoid is this disc-like shape and it's found in the anatomy of the chloroplast. So in the membrane of the thylakoid is where basically the beginning of photosynthesis occurs. Here's where we can see the first example of something called light reactions or light dependent reactions, meaning they require light energy to operate. In this stage of photosynthesis, light energy is used to power electrons, which come from water molecules, H2O, and they power these electrons in order for them to cross an electron transport chain. As the electrons go through these proteins and these carrier proteins, they eventually go to NADPH. So, however, to make NADPH in the first place, NADP plus reductase must be used and turned into NADPH. So, after light dependent reactions, two things are mainly produced, and that's ATP, which hold energy, and then NADPH, which is basically a reduced electron carrier. So these two products are essential in order to operate in the Calvin cycle and help eventually make the glucose molecule, which is the main point of photosynthesis. So in the Calvin cycle, Rubisco incorporates carbon dioxide, as we know, to create two molecules called 3-PGA. These two molecules then proceed through to something called the reduction phase in the Calvin cycle and receive energy released from ATP and NADPH to make something called 3GP molecules. And we're familiar with these molecules as they eventually make our glucose molecule. So these 3PG molecules are basically the main building blocks to make a glucose molecule for the plant to use as food. So overall, NADPH is crucial as it basically donates its electron to convert carbon dioxide molecules into sugars. However, if NADP plus reductase was removed from all the thylakoid membranes, NADPH would never be made, and thus not enough energy or electrons would be available to make G3P molecules, and the plant would not be able to make its own food. Therefore, in the end, the plant would slowly die out due to starvation, and the cycle would not be able to operate effectively. So that concludes my answers to my what if questions, and thank you for listening.